Okay, so have you ever wondered how plants actually grow? I mean, we see them all around us, right?
Yeah.
But how do they go from tiny seeds to these massive trees or even just like a little flower? It's kind of mind-blowing when you think about it.
It really is. And what's even more fascinating is that unlike us, unlike animals, plants, they never really stop growing.
Truly.
Yeah, like we reach a certain size and then that's it. But plants, they just keep going and going. It's called indeterminate growth.
Indeterminate growth.
Exactly. It's all thanks to these special groups of cells called meristems.
Meristems.
Yeah, think of them like tiny little factories scattered throughout the plant. They're constantly churning out new cells, which are like the building blocks for everything, from roots to stems, leaves, you name it.
So that's how a redwood tree can grow so tall. It just keeps making more and more cells.
Exactly. It's like a never-ending construction project. And each of those new cells has the potential to become anything, any part of the plant.
So how do they know what to become? Like, how does a cell decide to be a leaf or a root?
Well, that's where differentiation comes in. It's the process of cells taking on specific roles and developing into distinct tissues and organs.
So it's like career choices for cells.
Pretty much. Some become the construction workers building the plant structure. Others are the chefs, photosynthesizing and making food. It all depends on the needs of the plant and the signals it receives.
Okay, so that makes sense. But what about, think about like a tree that's been damaged. Can those specialized cells ever change back to a more basic state, like stem cells?
You're on the right track. Plants have this incredible ability called dedifferentiation.
Dedifferentiation.
Exactly. It's like rewinding the clock, allowing those specialized cells to revert back to a more basic, more versatile state.
So a leaf cell could essentially unspecialize itself.
Yeah. And then it could redifferentiate, meaning it can take on a whole new specialized role.
Whoa, that's amazing. So like a tree that's been, damaged by a storm or something could use this ability to repair itself.
Exactly. And it's also how plants can form new structures like bark or even roots from cuttings.
So it's like they have this built-in repair system that can adapt and change at a cellular level.
Exactly. It's one of the things that makes plants so incredibly resilient. But there's another key player in this whole growth and development story.
What's that?
Plant growth regulators, or PGRs for short.
PGRs. I've heard of those.
Yeah, they're like the puppet masters behind the scenes, controlling all the intricate processes of plant growth and development.
Puppet masters. Okay, now I'm really intrigued. Tell me more about these PGRs. What exactly are they?
They're basically a diverse group of chemical compounds that act like signals, telling cells what to do, when to divide, when to elongate, when to specialize. They're produced in different parts of the plant and can travel throughout, sort of like a communication network.
So it's like a complex messaging system within the plant, with PGRs as the messages.
You got it. It's how the plant coordinates all its growth and development, making sure everything happens in sync.
That's incredible. So how many of these PGRs are there?
Well, there are 5 major classes, each with its own set of roles and responsibilities.
5 major players. Okay,
Yeah.
I'm ready to meet them. Tell me, who are these key players in the plant growth symphony?
All right, let's start with auxins. They're often called the growth promoters of the plant world.
Growth promoters, okay.
They're responsible for stimulating cell elongation, influencing root development, and even guiding the plant's growth in response to light and gravity.
So they're like the conductors, making sure all the plant parts are growing in harmony.
Exactly. But the discovery of auxins is actually quite a fascinating story.
Oh,
Yeah, it all started with Charles Darwin and his son Francis.
the Darwins?
Yeah. They were studying how plants respond to light and noticed that canary grass seedlings would bend towards a light source.
Like they were reaching for the light.
Exactly. And through a series of experiments, they figured out that the tip of the seedling was somehow responsible for this bending.
So the tip was sending a signal to the rest of the plant.
Precisely. And later on, a scientist named F.W. Went actually managed to isolate this growth-promoting substance from the tips of oat seedlings. He named it auxin, and that was a pivotal moment in our understanding of plant growth regulation.
Wow. From observing bending grass to uncovering a key plant growth regulator? That's amazing.
So what about the other PGRs? Do they have equally interesting origin stories?
They sure do. And in the next part of our deep dive, we'll explore those stories and delve deeper into the fascinating roles of each PGR in the grand scheme of plant growth and development. All right, so next up we have gibberellins.
Gibberellins?
Yeah, gibberellins. And they're pretty powerful growth promoters.
Oh, really?
Yeah, their discovery is actually linked to a rice disease called bakane.
Bakane.
Yeah, it translates to foolish seedling.
Foolish seedling.
Yeah, so basically these seedlings, they would grow abnormally tall and spindly, and often they would just topple over under their own weight.
Oh, so they're like the opposite of a bonsai tree?
Kind of, yeah, but not in a good way. They were just too tall and weak.
Yep. So a Japanese scientist named E. Kurosawa was studying this disease, and he found out it was caused by a fungus.
Interesting.
Yeah, and he even managed to replicate the symptoms by treating healthy seedlings with extracts from this fungus.
So the fungus was making the seedlings grow tall and spindly?
Exactly, and that led to the isolation of gibberellins, these powerful growth promoters that could make plants shoot up like crazy.
So a fungus helped us understand how to make plants grow taller.
That's right. It's pretty amazing how these discoveries often happen, right?
Yeah, totally.
Okay, so now let's move on to cytokinins.
Cytokinins.
Yeah, and their story starts with scientists trying to grow plant tissues in the lab.
In the lab?
Yeah, it's called tissue culture.
Okay.
But they were having trouble getting the cells to divide properly, even with all the right nutrients and everything.
So they were like stuck.
Pretty much, yeah. And then by chance, they found that adding extracts from some pretty unusual sources, like coconut milk.
Coconut milk.
Yeah, or even, get this, autoclaved herring sperm DNA.
Herring sperm DNA.
Really?
Yeah, I know it sounds crazy, but it worked.
Wow.
These extracts contain compounds that could stimulate cell division.
Ah, so the coconut milk and the herring sperm DNA had something in them that helped the plant cells divide.
Yeah, exactly. And one of those compounds was eventually identified named kinetin.
Kinetin.
Yeah, the first cytokinin to be discovered.
So the quest to grow plant tissues in a dish led to the discovery of cytokinins.
You got it. It's amazing how science works sometimes.
Yeah, totally.
Yeah.
So now, what about ethylene?
Ah, ethylene, that's the gaseous one.
The gaseous one.
Yeah, the only gaseous PGR, and its discovery goes back to the early 1900s.
Okay.
People noticed that fruits ripened faster when they were stored near burning kerosene lamps.
Really? Like the lamps were making the fruit ripen faster.
Yeah, it was the ethylene gas from the lamps that was doing it.
Interesting.
Yeah. So ethylene is a simple gas, but it has a huge impact on fruit ripening, senescence, and other aspects of plant development.
So it's like the ripening hormone?
You could say that. Okay, so we've got auxins, gibberellins, cytokinins, and ethylene.
And last but not least, abscisic acid.
Abscisic acid.
Yeah, or ABA for short. This one was actually discovered by 3 different groups of scientists around the same time.
3 different groups.
Yeah, one group was studying leaf abscission.
Leaf abscission.
Yeah, the process of leaves falling off.
Okay.
Another group was looking at bud dormancy. And the third group was investigating a growth-inhibiting substance in cotton fruits.
Wow, so 3 separate paths, all leading to the same discovery.
That's right. And it turned out that abscisic acid is really important for helping plants cope with stress.
Like drought or extreme temperature.
Exactly. It helps them survive tough conditions.
So we've got our 5 major players: auxins, gibberellins, cytokinins, ethylene, and abscisic acid.
Yeah, those are the big 5.
Each with their own unique role to play.
Right. But it's important to remember that they don't act in isolation.
Oh.
Yeah, it's more like a complex interplay between all these PGRs and the environment that ultimately determines how a plant grows.
I see.
So it's not just about the individual hormones, it's about how they all work together in response to the plant's surroundings.
That makes sense. So the environment plays a big role too.
Absolutely. And that's what we'll be exploring in the next part of our deep dive, the fascinating interplay between PGRs and the environment.
Okay, so we've talked about these plant growth regulators, these PGRs, and how they're like these tiny little messengers controlling plant growth. But it's not just them calling the shots, right?
Yeah.
Like the environment has to play a role too.
You got it. The environment's a huge factor. Like, think about light. We talked about how auxins make plants bend towards light, but light also affects other stuff, like when seeds germinate and when plants flower.
Right, like how some plants only flower in the spring or summer when the days are longer.
Exactly. It's like plants have their own internal clock, you know? They can sense the changing seasons based on how much light they're getting.
So how do they actually do that? Like, how do they know when it's spring?
Well, it involves these special pigments that are sensitive to light. And those pigments can trigger changes in PGR levels, which then affect all sorts of things, like flowering.
So it's like a chain reaction.
Yeah.
Light hits the pigments signal the PGRs, and then the PGRs tell the plant to flower.
Pretty much. And it's not just light. Temperature plays a role too. Some plants need a period of cold temperatures to actually flower.
Really?
Yeah, it's called vernalization. It's like they need a good winter chill before they can bloom in the spring.
That's fascinating. So it's not just about the PGRs themselves, it's about how they interact with all these environmental cues.
Exactly. It's a complex dance between internal signals and external factors. And get this, PGRs can even affect gene expression.
Gene expression.
Yeah, they can turn genes on or off, which influences all sorts of things, from cell division to how the plant responds to stress.
Wow. So they're like master regulators,
Yeah,
controlling not just what cells do, but also what genes get expressed.
exactly. And understanding how this all works, this whole interplay between PGRs, genes, and the environment is super important for agriculture and stuff.
Like we could use this knowledge to help plants grow better, even in tough conditions.
Absolutely. We could potentially increase crop yields, make plants more resistant to drought or disease, even control when they flower and fruit.
That's incredible. It's like we're learning to speak the language of plants.
Yeah, it's pretty amazing.
Well, this has been an amazing deep dive. From those tiny meristem cells to these powerful PGRs, it's incredible how much is going on inside every plant.
I know, right? And we've only just scratched the surface. There's so much more to learn about this amazing world of plant growth.
Well, that's all the time we have for today's deep dive. But to our listeners out there, the next time you see a plant, take a moment to appreciate all the incredible things happening inside it. It's a whole world of complexity and wonder, just waiting to be explored.
And as always, keep those curious minds engaged, keep learning, and keep diving deep into the fascinating world of science. Until next time.